Okay, so it should be up and running. We should be recording. And last time we went through some of these first slides, kind of an overview in metabolism. We talked about some things like, how the macronutrients interconvert. We talked about essential nutrients. We looked at some of the pathways on how we actually get energy from food. And I gave you that percentage, right? I said about what percent of our energy each day comes from Krebs and ETC? About 90%, which means those are really important aspects of energy metabolism. So we'll talk about it a lot. But a thing to keep in mind is, how does that actually happen? Well, when molecules enter into the Krebs cycle, they're oxidized. And as they're oxidized, those electrons go into ETC. So we get most of the energy from our foods from being able to use electrons from the molecules in the macronutrients. Right? So if we look up at the top, the macronutrients can be converted into pathways, right, where they hit these common points, which is called intermediary metabolism. And then once they enter into Krebs, essentially what we're doing is we're taking components from our food, oxidizing it, and then using it for electron transport chain. The exception to that is carbohydrate. We can get ATP from carbohydrate without needing those electrons. We get it directly from glycolysis. So if oxygen is limiting, we rely on carbohydrates, and that's why. So we talked about that. We went through some of these. That looks horrible on this projector. This projector's probably 10 years old, so. And we got to here, right? Here. Before we go into this one, let's look at an example of this. I said I was going to give you an example of, for example, this one. How some of these plasma membrane proteins are involved in signal transduction, or actually information transfer and hormones. So the example that we're going to use, I guess you could title this little diagram we're going to go through would be like insulin and blood glucose. So what's the main function of insulin? What does it do? Yeah, lowers blood glucose. Good. Where does insulin come from? The pancreas. So if we say, and I'm a little bit low on, this is the pancreas. In the pancreas, there are several cell types, including beta cells of the pancreas. They release insulin. And insulin goes into the blood. And helps reduce blood glucose. So one of the questions is, where does the glucose go? Right? So you go home for dinner tonight and you have a meal and you haven't eaten for a few hours and you're sitting there and you're eating your meal and you might be kind of tired when you start, and then after a while you eat your meal, and partway through your meal or after the meal you start feeling like you have a little bit more energy. You kind of feel like, oh, I'm recovering. Well, what's happening? Blood glucose is going up. And then insulin should be released. And then blood glucose goes down in its concentration. Where's the glucose going? Let's say this is our small intestine. Right? We have our food, goes through the stomach into the small intestine. One of the foods we have, we're going to say, is glucose. Glucose goes into the blood. Here's an interesting thing. How does insulin get released? some glucose goes into the pancreas. And when the pancreas receives the glucose, sorry, I'm going to have to adjust this. When the pancreas receives some of that glucose, the glucose is metabolized and forms ATP. You didn't miss much. And I'm recording it, hopefully. So now when that ATP is released, the ATP actually causes insulin to be released. And we'll talk about this in 329, the specific details of that. It's a nice system, though, because normally what should happen is when blood glucose goes up, some of it goes into the pancreas. It's metabolized to cause insulin to be released. So that makes sense, right? And now the insulin goes into the blood, and the insulin actually binds insulin receptors on cells. Have you talked about this in other classes? Anywhere else? So insulin binds receptors that are in the plasma membrane. So this is an insulin receptor. It's one of these proteins embedded. So you can imagine that it's actually a dimer. So there's two parts to this. And then insulin would bind the protein. The insulin doesn't go into the cell. The insulin binds to the receptor, and then the receptor responds. And we don't have to remember this yet, but you've heard of phosphorylation of proteins and phosphorylation kinase cascades, all of that. Well, the insulin receptor actually autophosphorylates, which is pretty interesting. Insulin binds, the receptor phosphorylates itself, and that starts a signal. So we can just do this and say, it starts signal transduction. So the receptor has started a signal, and that signal basically goes inside the cell. And it causes this to happen in the cell. This little molecule right here is a protein. So GLUT4 equals a glucose transporter. And there's a family of these transporters, and one of them, the fourth one that was discovered, they just called GLUT4. So this is part of the Golgi. This will make more sense when we talk about more things in the cell coming up. You've heard of the Golgi apparatus, all right? So what happens is GLUT4 is made inside the cell, and the cell puts it in the membrane of Golgi, and they sit in the cytosol. And then when insulin starts this signal, part of this signal, one of the responses is this vesicle moves to the plasma membrane. And so now GLUT4 is in the membrane. Well, it's a glucose transporter. So this is what happens. Glucose goes inside the cell. And now glucose is coming out of the blood. Guess where most of that glucose goes? In the muscle cells. And now this glucose could either go through glycolysis, or it could be stored as what? Yeah, glycogen. Right? So glucose goes in here. That causes blood glucose to go down. Some of it goes into the pancreas. So we'll talk about this later, but this happens pretty quickly. Like if you were to drink pure glucose, like next quarter in the lab, you can. and then we'll measure your blood glucose before you do that and after. You'd see if you drink a glucose solution, within like 15 minutes, blood glucose starts going up. Not too long after that, that glucose will come back down. That insulin is sitting in the beta cells waiting for glucose to stimulate its release. So the whole system happens pretty quickly. And in general, it happens over and over again every day if everything is working correctly. If something's not working correctly and blood glucose stays elevated, that can be diabetes. So we'll talk about that term insulin resistance when we talk about diabetes in a few weeks. But that's what happens when insulin is released and this signal doesn't work right. That's what we call insulin resistance. So now what happens to blood glucose? Stays high. And depending on how long that occurs, a person can be pre-diabetic or become type 2 diabetic. Right? So here are the examples. We have a membrane protein here in the insulin receptor, and these are plasma membrane proteins as well. And if you were to characterize these, would you think that they are peripheral membrane proteins or integral? They're integral membrane proteins. I think that's it. Any questions on that? So this is kind of the level of details that you need to know so far for that. And I ask you to draw a diagram and explain that process. Okay, so let's look at the next one, this one, and then we'll take some of these details and apply it to this circumstance. Okay, so these are integral membrane proteins, a couple of them that are shown there, and these are how they're involved in transporting molecules across membranes. We're gonna look at three general transport mechanisms. So if I ask you to describe a transport mechanism, that's what I mean. It's diffusion, or it's facilitated diffusion, or it's active transport. And you guys have heard of these several times, probably since 10th grade or something. So let's go through it pretty quick. First one's diffusion, and this is simple diffusion. This is where molecules can pass through the plasma membrane. No ATPs required. It may not need a protein. So small, for example, small lipids can go through that without a transporter, because this membrane, that hydrophobic property in the middle of that membrane, allows little lipids to go through. May use a channel protein, that's what's being shown here, right? That's where it just kind of goes through the channel. There's no other mechanisms required. And it's concentration gradient dependent, right? We know what that means, right? It'll go from a higher concentration to a lower concentration until you hit equilibrium, and then transport across the membrane stops. So the next one's facilitated diffusion. The facilitator is a protein. So here's an integral membrane protein. Sometimes they show in books where you have like a protein that flips. Have you ever seen those? That doesn't happen. I'm not sure why they do that. But this is showing, what does this molecule look like? What could that be? 6-membered ring, oxygen bridge right there. Glucose, galactose, yeah. So in other words, GLUT4, facilitated diffusion. That's its mechanism. Right, it's concentration gradient dependent. So when glucose is high in the blood, that pushes glucose through GLUT4 until that concentration drops, and then it kind of hits equilibrium. But what the cell can do is, once the glucose goes in, it can modify it, and it'll trap it. So we'll talk about that quite a bit. So muscle cells can do that. Now this isn't the only tissue that it's going into, but it's the one that's insulin dependent. Right? And one of the advantages of this being insulin dependent is when glucose goes up significantly, it'll kind of focus its uptake into muscle, because muscle has GLUT4 and that relies on insulin. But the nice thing about this system is, let's say I exercise, and as I exercise, let's say I used a lot of the muscle glycogen. Now when I go and I have that post-exercise meal, if it has a lot of carbohydrate, that insulin helps me replenish that glycogen in muscle, because the insulin is allowing the glucose now to go back into that muscle and remake glycogen. And that energy source can be used for the next time I do high intensity exercise. You've heard of carbohydrate loading. And a lot of people think, oh yeah, I carbohydrate load, I got pasta the night before. It's more complicated than that. It takes like a week to do a successful carbohydrate load. And there's a little regimen we'll talk about later, but that's one of the advantages of having that there. Once the glucose goes in, the cell will modify it very quickly to glucose 6-phosphate, and then it's held in there. So even though it's facilitated diffusion, it's specific. So this will transport glucose, not glucose 6-phosphate. So it won't go back out. So we'll talk about that more later. And then so gradient dependent, requires a protein, no ATP required. Remember all that? Last one, active transport. What does the active mean? Needs ATP. The advantage is it'll run against a gradient. So in this case, this is showing the sodium-potassium pump, and no matter how much sodium is inside this cell, for example, on this side, more sodium can continue to do this. And we'll talk about that for carbohydrate absorption coming up. Calcium's another example of where that's really important. So, and there's a few different ways that these proteins rely on ATP, so we'll distinguish as we go through this. But remember that active transport requires energy, requires a protein transporter, and goes against a concentration gradient if necessary. So, glucose has to have a transporter in order to get into cells. This transporter is GLUT4. It depends on insulin. I said there's a few different family members of the glutes. Does it make sense if this one would be GLUT4 or another family member? Why?
Because there's no insulin to be
Good. That wouldn't work very well, would it? So this is actually another one called GLUT2 that allows it in. This is GLUT2. You guys will know the glutes really well by the end of this quarter. And so here's a good example. Rather than memorizing, just try to kind of think about how the body should do something, what makes sense, and a lot of times that's right. So in this case, can't have GLUT4 on that beta cell because that won't work. Another tissue that takes up that glucose is the liver. And we'll talk more about that later on, but the liver can also store glycogen. The liver does not have GLUT4. The liver has GLUT2, and we'll talk about why that's important there as well. So an example of proteins with slightly different structure and characteristics do the same thing, but they're on different tissues for a reason. And so we want to try to learn kind of the rationale on these things so that it's easier to learn and easier to remember. Okay, so let's talk a little bit more about that membrane before we move on. Went through this last time. We said, the membrane has mostly phospholipids, and a lot of those phospholipids not sure what I turned on there. Maybe light in another room. The phospholipids, remember, they contain 2 fatty acids, and one of the things to keep in mind is that saying, you are what you eat. You ever see somebody eating something, you're like, oh, you probably shouldn't eat that Twinkie, just way, RO3. And here's the reason why. Your dietary intake of different types of fats affect the type of fatty acids that are on your phospholipids and cell membranes. So for example, if you switch from a diet that is a lot of saturated fat to a diet that has a lot of unsaturated fat, that means more of our dietary fat has double bonds in the fatty acids, which means you now have plasma membranes that have higher amounts of fatty acids that look like this. Remember, these double bonds in nature are mostly cis. And when they are in the cis orientation, that means that they typically have a kink or a bend in that. If those fatty acids are part of phospholipids in a lipid bilayer, that causes those phospholipids to push apart slightly. Because now you have a fatty acid that will push the other fatty acids adjacent to it farther away from it. So when you compare this membrane, let's say a person doesn't have any unsaturated fatty acids in their membranes, it looks like this. All the phospholipids can pack tightly together. And now imagine that they have a lot of unsaturated fatty acids. The phospholipids are farther apart. That affects fluidity of the membrane, right? Which means that membrane is now more permeable. More molecules can go through it both ways. Which is better? What do you think? Has anyone ever asked you this? I heard saturated fat's not good for me. Should I consume saturated fat? They ask you this at Thanksgiving or the last part. Right? I just get that all the time. Anytime our family ate it, should I be eating this? You know, at some point you gotta eat something, right? What do you think? Should someone have saturated fat?
Like a balance, like a little bit of both.
Yeah, that's spot on. Yeah, you kind of want both, right? You want a good balance, and in this case, we probably want to eat about a third or 25% of our fat saturated, and the rest of it unsaturated. All right, we want some unsaturation and some saturation. So an easy way to say this is, we kind of want lipid bilayers with optimal characteristics or function, and we'll talk about that. But one disadvantage of the unsaturated fatty acids, they're less stable. The more double bonds you have, the more of those bonds can be oxidized or affected in some way that causes them to be destroyed. So they're less stable. So we'll talk about how that affects cell function and heart disease this quarter. So again, we kind of want a balance of saturated and unsaturated. How about this one? Here's cholesterol. It's hard to see. Space-filling model of cholesterol, right? We said it has, it's got a little, here's the structure of it. It's got ring structures. And when you see ring structures in a molecule, you usually say, oh, I think that's hydrophobic or hydrophilic. Mostly hydrophobic, right? And in this case, it's rigid, right? There's not any flexibility in those rings. So this part of cholesterol, hydrophobic. But look what it has on the end. That's an OH group. So it's got a little bit of a polar group here. So that means when cholesterol sits in that membrane, it sits so the cholesterol, so that hydroxyl group is toward the outside. So that's one thing to notice. But here's the ring structures. It said they're rigid. So in general, we're going to say cholesterol makes membranes more rigid, less permeable. Cells actually monitor that, and they can change the concentration of cholesterol in membranes to try to obtain a reasonable concentration in there. I've been to some seminars on this where it's fascinating the way cells actually determine cholesterol concentration in the plasma membrane especially, and then use cholesterol synthesis in the cells and cholesterol uptake from the blood to regulate that. So it's something that's usually well regulated. So with all of that said, we talked about last time, we said how much of this cholesterol in our body is from our diet? About a third. About 1/3 of our body's cholesterol from our diet, 2/3 from our synthesis. Okay, so that should tell us it is an important molecule. We make enough so we don't have to eat it, but it's important in our body. So the bottom part of it. So if we say decreased fluidity, that's like saying decreased permeability, right? The membrane is less flexible. All right, done with the membrane. Let's move inward into the cytosol, also known as the cytoplasm, the cytoplast. I'm fine with those interchangeably. I'll almost always say cytosol, probably because it's shorter. Okay, so inside the cells, we have the cytosol, and we also have a cytoskeleton. Those combined equal the cytoplasmic matrix. Cytosol is the fluid phase. It contains molecules like glucose, amino acids, oxygen, carbon dioxide, lots of other molecules. The cytoskeleton is the solid phase, or the polymeric phase. It has a few different components, and those include microfilaments. You've heard of those before. Those are actin chains. That's why actin is such a common cellular protein. You have microtubules, which are composed of a protein called tubulin. We'll look at that in a second. And then, right, actin's famous kind of counterpart, myosin, right? Not just in muscle cells, but it's in high amounts in muscle cells. And they make that intracellular kind of support structure. So some of the things that it does is it's a scaffold for the attachment of macromolecules. We'll look at that in a second. But molecules attach to it in order to affect cellular processes. It's also been shown that it's a site of localization for enzymes and substrates. When they first started characterizing the cytoskeleton, they realized, for example, glycolytic enzymes arrange themselves along the cytoskeleton to improve the efficiency of glycolysis. Right? So some pathways have their enzymes attached to the cytoskeleton close to each other so that metabolic pathway is more efficient. And you can just imagine, let's say, for example, glucose goes in, it becomes glucose 6-phosphate, and then it's got to wander around inside the cell to find the next enzyme to move down glycolysis. The cell can actually arrange those pathways physically along that cytoskeleton. So here's actin, for example. Those are actin subunits. Forms this kind of little small beaded structure. And then you have tubulin, which make up microtubules. Right, that's that hollow one, that protein that lots of proteins can use to move down. And then the last one, there's myosin. Probably seen that one before, right? It's got head, neck portions, and a tail, and basically that's a kind of a connective protein, solid structure. If you look at particular cells, you can actually do this. It's a very cool time to be in nutritional science, and we're in a pretty new area. And I even remember when I was an undergrad, I was working in a lab, and it's actually in the old chemistry building, which is probably still there. This particular lab, and I was in there doing these little plates, and they were called octoloni plates because they were an agarose gel, and we were actually making antibodies in chickens. So the protein chemist that I was working with a nutrition professor that I was working with, and they were working together to make these antibodies. And I used to actually have to go out there and get blood samples from the chickens after I caught them, take their blood, put it on this octoloni plate, and I could tell the titer of that animal's response to the protein. And I could tell when it was time to get their eggs, because we would get their eggs and actually spin down the eggs and get antibodies from that. And then we would get antibodies from their blood as well, so that we could use the antibodies to cure a specific protein. So I'm sitting there doing this, and all this stuff's going on in my head. I'm doing this, and I remember listening to this conversation where the researcher in the lab was talking to my professor, and they were going, "Oh yeah, molecular biology is now meeting nutrition." I'm like, and they're talking about all this. I have no idea what you're talking about. I don't understand this plate. And then I go to grad school, and I'm going, that's what they meant. So the beautiful thing for us is all of these other disciplines that have some overlap with nutrition, like biochemistry and genetics and everything, a lot of it has to do with areas of our health research that garners lots of money and support. So they develop these techniques that we can use in nutrition to actually study more specific things to nutrition. For example, it's to the point now where you can take a cell and you can take those cells and you can fix them to a slide. And this is a particular cell type called endothelial cells. And in your notes, you should start separating cell types. So endothelial cells are cells that make up, for example, walls of capillaries, or blood vessels, let's say. And they do that by connecting together. So they make extracellular proteins that actually attach to each other like this. And they'll do this, you can imagine, in kind of a three-dimensional structure. So what that means is they're attached to each other to be able to contain most of the components of blood, but not all of them, because nutrients have to be able to leave the blood, right? So they do that by making these proteins that are attached to each other. And they do this, and if you grow these in the lab, you can actually see the way they start attaching, and they'll form these more complicated structures. But what this is showing is these cells, see the way they reach out? They're reaching out with some of the attachments so that they can attach the cells and make structures that they require. You can look at the cell morphology by using antibodies that fluoresce, and the antibodies are specific to different proteins. Like anywhere on this slide where you see red, that means that this cell, these antibodies have attached to actin. And now when fluorescent light of a specific wavelength is shined on this stain, you get a red color. Antibodies that have a green fluorophore attached recognize tubulin, and that's the microtubules that you see. They also stain the chromatin, the nuclei, blue. So you can see that these cells have elaborate structure. And so these obviously are eukaryotic cells. So one of the things that we need to keep in mind is we use a significant portion of the energy from the food that we eat every day for doing things like making cells. If we don't take in the right nutrients, it compromises things like this. It can affect our health in very negative ways. It can cause lots of different types of ailments based on deficiencies, because all of this complex, these systems that we have require specific nutrients in adequate amounts. So it has a lot of effects in our body. And so we just think, when we think about those cells, we need to think about that. The other thing that we should think about is all of the pathways that we're going to talk about occur in specific parts of the cells. And you guys need to remember where they happen in the cell. And the reason is because that involves the regulation. Glycolysis is in the cytosol for a reason, lots of reasons. Krebs cycle is inside the mitochondrial matrix for lots of significant reasons. So we want to remember all those as we go through this. So we're going to start in an overview of metabolic pathways and where they are by looking at the pathways that are in the cytosol. So let's start with a basic definition of glycolysis. We're going to say glycolysis is the metabolism of glucose to pyruvate. More specifically, how many pyruvates? 2, right? Glucose has 6 carbons. Pyruvate, 3. So we make 2 pyruvates from each glucose. Right? Glycolysis in the cytosol. Glycogenesis. Right? That is the conversion of glucose to glycogen. So glycogen is all glucose. It's the storage form of glucose in animals. And in our body, where does glycogenesis occur? Which 2 tissues? How about one? Who can give me one? Yes, muscle. The other one's liver. In 328, if you're not sure, say liver. It's metabolism. Actually say, if it's tissue, say liver. If it's molecule, say glucose. You'll have a good chance of getting it right. Glycogenolysis is the opposite. Remember lysis? That means to break. So this is going glycogen to glucose. So you can store that glucose and get it back. That occurs in the cytosol. As a matter of fact, if you have liver cells and you take them from an animal that was just fed and they have glycogen stored in the liver, if you set up your microscope just right, you can see these little glycogen granules within those liver cells. If they fasted for a long time, you won't see those granules because they've used the glucose from their liver to maintain blood glucose. It's in that cytosol. Does that make sense? Because now if we want to go from glycogen to glucose to glycolysis, it's all in the cytosol. Fatty acid synthesis. We're going to briefly define this as fatty acid synthesis is the synthesis of fatty acids from acetyl-CoA. And we kind of looked at that before, right? We can see it. You can't see that very well. Here's acetyl-CoA. Here's the fatty acid. That happens in the cytosol. And there's quite a few places in the body that can make fatty acids. We'll talk about that later. Okay, let's move into mitochondria. My favorite organelle, right? Does lots of different things. Right, so I'll go through this pretty quickly. You know, the basic structure of the mitochondria is 2 membranes. Outer membrane, inner membrane. So in your notes, outer membrane, relatively porous. What does that mean? Yeah, it's permeable. Quite a few molecules can go through that outer membrane, no problem. No transporter needed. Inner membrane, non-porous. Very stringent per the passage of molecules. Nothing goes through that inner membrane that shouldn't. If it does, mitochondria is destroyed. It's dysfunctional, the cell will remove it. Well, what's one of the reasons that's so important? What's on that mitochondrial inner membrane? ETC, right? Those proteins are embedded in this inner membrane. That inner membrane is so tight, not even what can go through it? Protons. And what's a proton? I ask a lot of questions. I just keep digging in, right? We'll drill down. What's a proton? Where's it come from? Remember that? I fell for it again. That's a proton. Means nothing. That's an electron. What's this? It's the simplest element. Look on that periodic chart, upper left. That's hydrogen. So ETC separates the electrons from the protons of hydrogen. Protons move down that gradient. Sorry, electrons move down the gradient as protons are pushed out across that. And then the protons will hang around the outside of that inner membrane, try to get back inside. Their only way back in is your ATP synthase. And that's what drives ATP synthesis. And you can look at it like it's all dependent on the non-porous characteristic of that inner membrane. All right, what else? In between the outer and the inner membrane, very important space, that's called the intermembrane space. It's not intermembrane space. I don't know where that is because of that. That's intermembrane space is in between, and that's where that gradient's established, right? What else? Oh, the matrix. Oh, the crystal. Let's do that. So kind of your notes for the inner membrane. Not only is it very non-porous, it has crista, and the crista are the increased surface area of the inner membrane. Remember that? They're like folds. The more surface area you have, theoretically, the more ETC components you can embed in that membrane. So it makes it more efficient. Let's go all the way inside, talk about the matrix. What pathway is inside that matrix that we've talked about a few times already? Krebs cycle is in the matrix. There's lots of other things inside that matrix, including if you do a mitochondrial prep, for example, before I came here, I was working on research, and we were using yeast as a genetic model. And a lot of people say, "Oh, power of yeast genetics." Well, I hated them because their genetics could change so quickly; they'll outsmart you. And what you do is you start out with these big containers of yeast, and you inoculate the containers, and it's some type of solution that you put the yeast in. And then you agitate them, you aerate them so that they can generate plenty of energy. And so what you do if you're going to do a mitochondrial prep, what we were doing is we were altering the genetics of a protein that was embedded in the mitochondrial inner membrane. Because people that had this genetic condition used to have abnormal mitochondria. They used to develop what's called sideroblastic anemia, which means their red blood cells did not function properly, and it caused significant physical effects. It altered development, or altered their ability to move around. It was not a harmless condition. So we were characterizing how this protein works. So we used to make changes in the genetics to match those of people that had the condition, and then we'd figure out what was going on with the protein. And we were able to figure out what one specific abnormality did. But as part of that, what we would do is you'd grow up these large colonies of yeast that had your genetic alteration, and then you'd isolate the mitochondria. So the day before, like about 3 o'clock in the afternoon, I would start my cultures, and I would grow them in like these 3 large 1-liter flasks. Next morning, I'd come in and I'd start pouring my solutions into these bottles, and I'd spin down the yeast. I'd collect this big yeast pellet, and then I'd start breaking down the wall of the yeast so that I could get to the interorganelles, including the mitochondria. Long story short, in the early afternoon, like at 2 or 3 in the afternoon, I'd have mitochondria in my tube. I'd go through like these 2 or 3 gallons of solution, and I'd end up with one small little pellet on the side of my tube. Then I would take a buffer and I'd resolubilize that. I'd take the pipette tip and I'd pipette it up and down. Then if I took my pipette tip out, I could see that strand of looked like silk. What is that? DNA. A mitochondrial DNA. So in the matrix, they have DNA. What's the DNA in there for? Transcription. Right, so inside the matrix, they have DNA. Right, they can express those mitochondrial genes to make proteins that are mostly involved in what? ETC. Most of the mitochondrial DNA expresses ETC genes. In other words, proteins that are required for ETC. If you can make those proteins, you can do transcription and what? Translation. Remember, transcription is DNA to RNA. Translation is RNA to protein. All that happens inside the matrix. Now you have a protein that's an ETC, it can embed in that membrane, and it helps ETC function. So that's what all that does. The other thing that you'll notice when you're solubilizing that and you get this solution, it looks like you got dirt in there. It's kind of a dirty, rusty color. What's that? What's that rust color? What's the most significant nutrient deficiency in the world? Deficiency of what? Iron. Iron. Huge percentage of the population. Classic symptoms of iron deficiency. Lack of energy. Impaired development. Lack of cognitive abilities. What does it do? Compromises energy production. Why? Those ETC proteins, a lot of them use iron to transport the electrons. So there's a lot of iron inside the matrix and in that ETC membrane. That's why your solution turns that kind of brownish color. These individuals with sideroblastic anemia, they used to accumulate about 40 times more iron than most people. And that's what was kind of toxic to this situation. That iron was not being released to be used in the cell for other things. And so it was a significant alteration. And it was actually a transport protein that was altered that couldn't move that iron out. That's what us in another lab figured out. All right, so lots of stuff going on in there. Here's the thing. Primary site of oxygen usage. Why? ETC is required. Oxygen is required for ETC. So it's the powerhouse of the cell, but most of the ATP that's made in the matrix isn't used in the matrix. Right? ATP is made in the matrix, transported out. We'll look at that in a few minutes. Outer membrane. I think I went through all this. I did. Oh, the last one. Remainder of mitochondrial proteins, those that aren't encoded by the DNA in the mitochondria, are made in the cytosol and then incorporated in the mitochondria. That's what the last one means. That's most of the mitochondrial proteins, but. All right, so here we go. Metabolic pathways in the mitochondria. Krebs cycle, also known as TCA and citric acid cycle, right? Hans Krebs cycle. Electron transport chain is there. So if I ask you where the electron transport chain is, you're going to say mitochondrial inner membrane. Fatty acid beta oxidation. How would you summarize that? It's the oxidation of fatty acids and production of acetyl-CoA. Sure. Fatty acid beta oxidation is the oxidation of fatty acids and synthesis of acetyl-CoA. And then the last one is heme biosynthesis. Where have you heard that name before? Heme. Heme of what? Hemoglobin. Right? So hemoglobin is when globin, the protein, wraps around heme. Remember that structure of heme? You guys saw that in other classes? Heme is basically like a flat cyclic molecule. It's called a protoporphyrin 9 ring structure. Once the mitochondria adds an iron atom to the middle of it, it goes from protoporphyrin to heme. And then it's released from the mitochondria so it can be used for other things like making red blood cells. So that heme biosynthesis is there because all that iron is there. And anything that impairs mitochondrial function can affect that, and that obviously can affect red blood cells. So the mitochondria are extremely important for lots of things. So what cells in the body have mitochondria? Almost all of them. The exception, red blood cells. We talked about that last time. So here's an overview of what we just talked about. Right? So let's imagine this is a cytosol, and so let's just say here's glycolysis, right? Glucose to pyruvate. Then pyruvate goes across the mitochondrial membranes and into the matrix where it's converted to acetyl-CoA. Pyruvate can do this. It's got a transporter. It can go across the membranes. Once we make acetyl-CoA, it's trapped. It's got to come out as a different molecule. Commonly, it goes into Krebs cycle. But see, acetyl-CoA can either come from glycolysis or fatty acids. We'll talk about that later. Once we have Krebs cycle, we're generating CO2, providing electrons. Here's our little simple diagram of ETC. And here's oxygen being used to accept the electrons to make water. Not a complete reaction, or missing protons here. But anyway, and then one of the points is, these protons go back in. This is what? ATP synthase. And then here's ATP. Some could be used in here. Most of it transported out. This is transporter for it. Goes out. What else was I going to say? I think that's it. Oh. So here's beta oxidation. So here's an important point. Fatty acids being converted to acetyl-CoA is fatty acid beta oxidation. Acetyl-CoA to fatty acids, that's fatty acid synthesis. That's in the cytosol. The cell puts synthesis and oxidation, two different parts of the cell. They have common molecules. So what the cell does in order to keep those from running at the same time and becoming a futile cycle, separates them. That's an advantage of that. Right? Because in general, if a cell is making a lot of fat, it's not oxidizing a lot of fat. Good there? And this just shows, again, another slide showing some of the characteristics. Mitochondria, these two cells, here's one cell. Mitochondria in red, they tend to be distributed throughout a cell. If this was a muscle fiber, they'd be squeezed in between those filaments. And then the Golgi's green, we'll talk about that in a second. And then this is obviously is the nucleus and the chromatin stained with the blue. And an interesting part for mitochondria, they're very dynamic. They'll stay around for a relatively short period of time. It's usually a few days, and then they're destroyed and replaced. So mitochondrial numbers can change a lot. It's called mitochondrial density in tissues. So for example, if I go from not doing any exercise, and if I was to quantify the mitochondria, the muscle fibers at that point, they might be relatively low. Now, if I start an exercise program, how long will it take for those mitochondrial numbers to start going up? Like a day. It'll start almost immediately. Hypoxia and some of the other conditions will stimulate mitochondrial biogenesis. So that's the good news. Now I stop exercising. How long before they start going away? Almost immediately, right? Use it or lose it. Right? So my mitochondria are very confused. Right? But you can quantify that. You can take a muscle fiber and basically quantify that before and after as an indicator of mitochondrial function. And here's kind of an interesting thing. There's certain parts of exercise that are absolutely dependent solely based on mitochondrial function. In other words, you can't go anymore. You can't produce any more energy because mitochondrial numbers and function are too low. That's one of the things that can stop a person from exercising or performing physically at a really high level. It's mitochondria. It's very well known. So that's something that we have to support with good nutrients and a good exercise regimen in order to maximize mitochondria. They're that important for energy production. Good there? All right, so let's take a break. I'll stop the recording here, and then we'll continue on with lysosomes in a minute.